In our last video, we talked about some basic classifications of terms that we can use to describe organisms and the cells they contain. I am a human being, an animal, so I would be a multicellular eukaryotic organism, meaning that I have many different kinds of cells that make up me as the organism, and then each of those cells would be eukaryotic, which means that they do contain a nucleus and they do have membrane-bound organelles. Whereas if we were to take a culture of bacteria on my skin, we might find that we have unicellular prokaryotes living on my body. Unicellular means they'd be a single cell, and prokaryotic would mean that the cell, and in this case the bacteria, would lack a nucleus. They would not have a nucleus or membrane-bound organelles. So an easy way to remember this is prokaryote is the no karyote, and eukaryote is you. Or you can think eukaryote is the do karyote. So it does have a nucleus and they do have membrane-bound organelles. And here is just a little classification chart to help you remember which organisms belong to which category. Now, one thing we do need to remember is that there are things that all cells have in common. It doesn't matter if they're prokaryotic or eukaryotic. So you can see here in this diagram, we have our prokaryotic cell and our eukaryotic cell, and I have highlighted some of the similarities or things that these two cells have in common. And one of which is the cell membrane. And remember, the cell membrane is a selectively permeable phospholipid bilayer. And I know that's a lot of scientific vocabulary to juggle around, but selectively permeable means it simply selects what can permeate through the membrane, and permeate means to pass through. And so it can kind of regulate or control what enters and exits the cell, and that's important for maintaining homeostasis. And the phospholipid bilayer just refers to the fact that it contains lots and lots of phospholipids, and there are two layers of them. That's where the bilayer comes from. Now, the cytoplasm refers to everything in the inside, that jelly-like fluid and the other things floating around. And you will sometimes see the term cytosol. That was just simply the solution found in the cytoplasm, right? So it's aqueous, so it's water-based, and you've got lots of things floating around. All the organelles are suspended in it, and it's also a place where reactions can take place. We learned about cellular respiration. We know that glycolysis takes place in the cytoplasm. And if the cell is relying on, alcoholic fermentation or lactic acid fermentation, that will take place in the cytoplasm as well. And you can see in both the prokaryote and the eukaryote, we've got these tiny little dots, right? And these are ribosomes, right? Ribosomes are the site for protein synthesis. So you can think about a ribosome like a protein factory, and they're not true organelles, right? So there's no membrane covering around a ribosome. We'll later talk about what ribosomes are actually made out of. They're made out of RNA and proteins combined together, but it's a protein synthesis site, right? It's a protein-making factory, and both prokaryotes and eukaryotes have them. Prokaryotes have a different kind of ribosome. It's the 70S ribosome, where eukaryotes typically have an 80S ribosome. And then last but not least, both cells are going to have DNA, right? So the instruction manual for life. And DNA is a molecule that contains codes, and those codes are for specific proteins, okay? So humans have unique codes for proteins that make us human beings, whereas plants have unique codes for the proteins that make them plants. And all cells will actually, believe it or not, have some of those codes that will be similar. Prokaryotes have circular DNA, whereas eukaryotes have linear DNA. That's another distinguishing factor, but both at the end of the day have DNA. Now, let's talk about eukaryotes, right? And the organelles that they contain, right? So organelles, you just think like baby organ or tiny organ. So in the same way that our bodies, we have many organs that perform specific functions, cells or eukaryotic cells, such as the one I have here in this diagram, have many different organelles. And each of these organelles has a different function. And this allows the eukaryotic cell, right, to compartmentalize certain tasks, right? And so in one organelle, you can have specific enzymes that are performing a certain job, and in a different organelle, you might have different enzymes and different molecules performing a different chemical reaction. If we didn't have these organelles, all of these enzymes would be floating around, and we wouldn't be able to control where reactions were taking place, and it would be a little bit more chaotic inside of our cells. So let's talk about some of the organelles that make eukaryotes so unique. and one of which is the nucleus, right? And you can think of the nucleus as like the control center or the brain of the cell, right? And so the nucleus has its own membrane, right? And so inside of the nuclear envelope or the nuclear membrane, we're going to find DNA. And usually, under normal operating conditions, our DNA is going to be wrapped around some histone proteins as nucleosomes, and we refer to those wound coils as chromatin, right? And so typically our DNA can be found as chromatin. And inside of the nucleus, we can also find something known as the nucleolus. And the nucleolus is a unique part of the nucleus. It is responsible for synthesizing ribosomes. So if you remember from the last slide, ribosomes are the little protein-making machines. Well, they're actually synthesized inside of the nucleolus. Now, the nuclear membrane helps protect DNA from viral infections and from other pathogens, and then also from, oxidation molecules that could disturb or change the structure of DNA or could damage our DNA. And so our DNA in eukaryotic cells is protected by not just the nuclear membrane, but also the cell membrane. Okay. And there are nuclear pores. There are small little openings that can allow certain molecules to enter and exit the nucleus. And those nuclear pores or those openings will become very important later on in the semester when we start talking about transcription and translation. So let's go ahead and move on and start talking about the mighty mitochondria. And so we know all about the mitochondria now. We know it's the site of aerobic respiration, right? And so glycolysis would take place in the cytoplasm. And then our molecule of pyruvic acid would be modified into acetyl-CoA, and then it would eventually end up in the matrix, this yellow portion here, where the citric acid cycle would produce lots of molecules of NADH and FADH2, which will be used in the electron transport chain to produce lots and lots of ATP. So this is the ATP factory, if you will. Now, what's really neat about the mitochondria is the mitochondria has its own DNA. Right? And it has its own ribosomes. So the mitochondria actually can self-replicate, right? So within our cells, our individual mitochondria can make copies of themselves depending on different environmental stimulus. And the DNA inside of your mitochondria is actually, it can be used to trace your maternal lineage. In other words, you got your mitochondria from your mother because they were present in the egg that was fertilized by your father's sperm, right? And so if an individual were to have some kind of mitochondrial disorder, their mitochondria wasn't functioning properly, it could be traced back through their mother's family history. And later on, when we talk about genetics and genetic disorders, we'll talk about some of those mitochondrial diseases. And then what's really neat about the mitochondria is its ribosome. The mitochondria have 70 S ribosomes, as well as circular DNA. If you think about that for a minute, you think, wait a minute, circular DNA and 70 S ribosomes, aren't those characteristics of prokaryotes? And so that's where, one of the bases or one of the basic ideas that lays the foundation for the endosymbiotic theory. And so a lot of scientists and a lot of evidence supports the idea that the mitochondria may at one time have been its own prokaryote, its own cell that became endocytized or started living inside of another cell. And you might think that's ridiculous. So a cell living in another cell, but we actually find it in nature quite often, right? So these mitochondrial organelles may have at one point been their own organism. And so we consider the mitochondria an autonomous organelle. And the chloroplast would kind of fall into this same category, right? So the chloroplast would be the site of photosynthesis, right? So we know that photons hit chlorophyll that are embedded in the thylakoid membrane, and that excites an electron, and that electron zips across some electron transport proteins, pumps hydrogens, and creates a gradient, which we can then use to form ATP, and that electron eventually forms NADPH, which carries the electron over into the stroma. And in the stroma is where we have the light-independent reaction, where we finally fix carbon dioxide into glucose. Now, the chloroplast also has its own DNA and also has its own ribosomes. It's also capable of self-replicating, and so it also fits into that endosymbiotic theory, and that the chloroplast may have been at one point its own photosynthetic prokaryote. And we do see some bacteria that still perform photosynthesis, and it's a little different than the photosynthesis that we see inside of eukaryotes, but it's very similar in structure and to what happens inside of a chloroplast. So these may have at one point been their own organisms. Now they live or reside within photosynthetic eukaryotes. And then we have the endomembrane system. So in science, when you see endo, right, just think inside, right? So this is an internal membrane system, right? So you can see the nucleus here, and you can see these highly folded membranes that kind of extend off of the nucleus, and they actually reach all the way to the plasma or cell membrane. And there's different parts to this endomembrane system, and each of these has a unique function, right? And so if you look right here, you can see the rough ER, and we call it the rough ER because it's studded with ribosomes, right? And so the rough ER is rough because the membrane contains ribosomes. And if you remember from the earlier slide, ribosomes synthesize proteins. And so the rough endoplasmic reticulum, that's what the ER stands for, endoplasmic, and endo means inside, plasm referring to the cytoplasm, and reticulum I think is a Latin word for net or to fold, right? So if you think about a reticulated python, right, the way they kind of fold back on themselves, you can see that there is a reticulated pattern here, lots of folds, right? And so we have our rough endoplasmic reticulum, and in the membrane, there are embedded ribosomes. So the ribosomes synthesize proteins and then dump them into the rough ER where they can be folded and assembled into their final form. And at some point, in some proteins, they'll actually be tagged with little carbohydrates. It's like little labels, like this protein does this, or this protein needs to go here. And so cells that assemble and produce a lot of proteins are going to have a lot of rough endoplasmic reticulums, right? And so the cells in our pancreas that are responsible for producing insulin and glucagon, which are protein hormones, they're going to have a lot of rough endoplasmic reticulum. And then you've got the smooth ER, right? And so the smooth ER would be any portion of the endoplasmic reticulum that lacks ribosomes. And the smooth ER is responsible for producing lipids, right? So producing fats, oils, and waxes, and steroid hormones, right? So remember, steroid hormones would include things like cholesterol, testosterone, and estrogen. So cells that are responsible for producing lots and lots of lipids would have lots of smooth ER, right? So our testes and our ovaries, right? The organs that are responsible for producing testosterone and estrogen, they're going to have a lot of smooth endoplasmic reticulum. The smooth endoplasmic reticulum is also very important for detoxifying compounds. So the human liver, the organ responsible for detoxification, is going to have a lot of smooth ER. So if your liver becomes damaged, say from repeated use of alcohol or other drugs, you could compromise your body's ability to metabolize, or in this case, catabolize, break down these toxins. And a lot of those problems would be associated with maybe a lack of function with your smooth endoplasmic reticulum. And an easy way to remember this, I think smooth, I think lipid, I think smooth like Buddha's belly, right? And so, big fat belly is gonna be nice and smooth, and it just helps me think, okay, smooth ER, we're talking about lipids. And then the last thing we have here is the Golgi. It's a really funny name. It's a last name of a scientist who discovered this, is what he's most famous for, but he also did a lot of other really amazing things. But the Golgi apparatus or Golgi complex is kind of like the post office of the cell. So as the rough endoplasmic reticulum, folds and puts the finishing touches on proteins, and the smooth endoplasmic reticulum makes lipids, they end up here in the Golgi. And the Golgi apparatus or Golgi complex will kind of pack and label and ship these where they need to go. And so typically speaking, proteins that are folded and assembled in the rough ER, their ultimate destiny is probably going to reside somewhere on the outside of the cell, whether it's embedded in the plasma membrane or it'll be secreted and released into the extracellular fluid or the fluid outside of the cell. And so secretory cells that secrete a lot of compounds, are going to have very, very active Golgi complexes or Golgi apparati. And you can see here there's a cis and a trans side to the Golgi. So the cis side would be like the receiving end, right? So the cis Golgi would receive the assembled proteins from the rough ER and the assembled lipids from the smooth ER, and the trans side would be responsible for the secretion, right? Finally releasing these vesicles, and a vesicle is just a little membrane-bound sac, right? So maybe inside of this little vesicle, we have insulin, right, which would have been assembled in the rough ER, and it would be released into the bloodstream to tell our cells, hey, blood sugar is too high. Let's tone it down a notch. All right, so let's move on to the next slide. Uh-oh, my bad. Little technical error there. Okay, and so we also have vacuoles. And as I had mentioned, vacuoles are like kind of modified vesicles. They're a little larger, right? And so there are different kinds of vacuoles in different cells. So plant cells have a large central vacuole, and that's something that's very unique for plants, this massive, large central vacuole. And so why do plants need such a large vacuole? Well, it doesn't rain every day. And so plants need to have a place to store water to perform photosynthesis. Remember, water is a key reactant in photosynthesis, and so plants need it to perform photosynthesis to synthesize glucose, and so that water can be kept in the vacuole. Now, plants also keep other things in the vacuoles, right? So the certain pigments that give flower petals their unique colors are going to be found in the vacuole. Certain plants that taste really bitter or that could be toxic, the plant is going to keep those bitterant compounds, usually they're alkaloids, in the vacuole. And there are also some enzymes in there as well to maybe break down certain compounds. But the primary constituent, or the thing that we find in the largest volume in vacuole will be water. And so it also serves another purpose. As the vacuole fills with water, it applies pressure, right? Pushing against the outside, right, portion of the cell, and it keeps it rigid, right? And we call that turgor pressure. And so if you haven't watered a plant in a really long time, you may have noticed that it starts to wilt and starts to kind of flop over on itself. And then you water your plant and the plant stands back upright. And that's because the vacuole fills with water and the turgor pressure increases. Whereas when the plant is dehydrated, the turgor pressure decreases and the plant might wilt. Some cells also have something known as a contractile vacuole. And we usually find contractile vacuole in aquatic protists that live in fresh water. These organisms, since they live in fresh water, have to continually control the amount of water rushing into their cells. Because if they don't have a way of controlling that, the cell could potentially expand and explode. So they have these contractile vacuoles so that as water enters the cell, they have a way of squeezing or squishing it back out to prevent them from exploding, which would not be good for those particular organisms. We also have two other really neat organelles, the lysosome and the peroxisome. The lysosome is kind of like a cleanup organelle, right? It has the ability to break down old organelles. It has the ability to break down bacteria or other pathogens or just other waste products that might find its way into the cell. And the lysosome contains a lot of hydrolytic enzymes. Hydrolytic simply means they perform hydrolysis. Now, these hydrolytic enzymes really prefer a pH of 5, right? And we know that that's really acidic, okay? And so the hydrolytic enzymes in the lysosome only function at that pH. And so if a lysosome were to become damaged and those hydrolytic enzymes were to leak out in the cytoplasm is not going to have a pH of 5. And so that instantly denatures those enzymes, which is important because otherwise those hydrolytic enzymes would, start catabolizing other molecules in the cell and can potentially dissolve the cell from the inside out. And so that's another example of the importance of enzymes and their preferred pHs. Now, lysosomes are also considered suicide sacs because they are believed to play an important role in what is known as apoptosis. And that would be programmed cell death. You might be thinking, why in the world would our cells want to program their own demise? Well, we'll talk about the significance of that later on. And then we have the peroxisomes. The peroxisomes are also little vesicles that contain important enzymes. And the peroxisomes are more important for lipid catabolism, breaking down lipid molecules. And the peroxisomes also help break down peroxides and other very damaging compounds that can oxidize molecules. Oxygen and oxygen ions are very reactive, and they can react with all kinds of molecules in our cells and interrupt or inhibit certain chemical reactions. And so the peroxisomes make sure to clean up those compounds and help protect the cell. This is another reason we need to eat antioxidants, right, to help prevent those oxidation reactions that could destroy our cells. And so peroxisomes are also going to be really important for catabolizing or breaking down alcohol. Now, typically we only find, and I don't want to say we only find, we find lysosomes in a lot of animal cells, but you might not see a lot of them in plant cells per se. Okay, let's continue marching along. And then here I have some structures. Okay, these aren't true organelles. These are not membrane-bound organelles. These are cellular structures, and one of which is the cytoskeleton. And so in the same way that we have a skeleton that provides structure and support and protection for our bodies, cells also have a cytoskeleton. Now, obviously, different cells are going to have different cytoskeleton structures, and it varies from one organism to the next. So obviously plants and animals are going to have different cytoskeletons. A fungus and an amoeba are going to have different cytoskeletons, but all these cells are going to have some form of cytoskeleton. Even in our own bodies, the cytoskeleton in your skin is going to be different than the cytoskeleton that you'd find in a muscle cell. Now, we also have something known as a cell wall. Now, humans and animals do not have a cell wall, but plant cells have cell walls, fungus have cell walls, and most protists have cell walls. And even some bacteria have cell walls as well. And they're made of a lot of different things. So we talked about cellulose earlier on in the semester. Cellulose would be what plants use to make their plant cell wall, which gives them an extra layer of protection and an extra layer of structure. Fungus, their cell wall is actually made of chitin, which is the same thing that you would find in the exoskeleton of a lobster or a crab. And we'll talk about how crazy that is later on in the semester when we start to dive into evolutionary biology. And then some cells have cilia, okay? And cilia would be tiny hair-like projections that are found on the outside of cells. And in a circular motion, they can allow certain eukaryotic cells to move, right? And so these cilia are only going to be found in eukaryotes, and they're like tiny little Viking oars, right? So they'll all be working together to allow this cell to move. Now, not all cilia are responsible for making the cell itself move. Some cilia are responsible for making the solution or the environment to move around the cell. We have cilia in the back of our throats that allow particles that get stuck to be knocked down into our digestive tract. Now, we all have probably sat in a movie theater before behind or in front of or around someone who smokes regularly. And the heat in the smoke can actually denature some of the proteins in the cilia. And if you smoke often or enough, different compounds can inhibit the production of the cilia. They can denature the proteins in the cilia, and they could even damage and scar those tissues. And since the smoker doesn't have the cilia to move those particles, they'll develop a smoker's cough. And then we've got a flagella. That'd be many, plural. Flagellum would be singular, and that is like a long tail-like protein projection. And so the most famous flagellum would be what you'd find in human sperm. Okay? You'd find that in human sperm. Now, we can find flagellum in lots of different kinds of organisms. Some bacteria have flagellum, and even some plant cells have flagellum, which is crazy that certain plants, their reproductive cells, can have these little flagellum. And the flagellum, or flagella, if we had many of these, also aid in locomotion, right? They provide a way in which these cells can move throughout their environment, which is really wild to think about. And they are like little motor proteins, right? And they'll be kind of moving in a whip-like fashion. And so how are you going to remember the difference between these two? Well, I think flagella, I see this flag right here, and it just makes me think of a flag kind of like flapping in the wind. And when I think of cilia, I just kind of think about the word. And, you've got the little lowercase I and the lowercase l. And if I write cilia along the membrane of a cell, the I and the L kind of look like those little hairs. I don't know. It worked for me, and it's worked for the last, well, almost 2 decades in being able to remember these.